Hello students, this is Faizan Mirza. We are discussing the male reproductive system in this lecture. The male reproductive system involves a series of glands, a series of tubules, and a series of external genitals as well. The term reproduction means how the new offspring can be produced and how the genetic material can be passed on from one generation to the next. Then the gonads are the region where the gametes are made, the sperm and the ova. These are testes and ovaries in males and females. This is the female reproductive shown to you, reproductive system shown to you. The male reproductive system shown to you. You can see the gonads as ovary and the testes labeled appropriately. There are various exocrine and endocrine glands which actually are working in the reproductive system. There are ducts in the reproductive system and accessory glands that produce material that support your gametes. The male reproductive system include the testes and the testicle. The testes are basically paired over glands measuring about 5 centimeter in length, 2.5 in diameter, and weigh about 10 to 15 grams. They are partially covered by a serous membrane called tunica vaginalis. There is basically an outpopping of peritoneum, and the internal to this tunica vaginalis is tunica albuginea, and this is this which extends to the inside the testes and divides the testes into various lobules. So this is the testes here. You can see it is the testes is having the head of epididymis here. This is the scrotal sac, and from here you can see the sperm duct goes high. It loops over the urinary bladder, it joins the urethra and comes out through the urethra into the external orifice. This is the external genital, the penis. It is having the region called the glans penis. There is the prepuce, the external urethral orifice, and the corpus spongiosum is also present, which we'll be discussing in detail later. Scrotum is basically a cutaneous outpouching of the abdomen. It is having loose skin and superficial fascia. It is supporting structure of testes. It allows the body temperature to be stayed. It allows a lower temperature, 2 degrees centigrade lower temperature than the body temperature, which is required for the maturation of sperm. The septum here, which divides it from the two, so you can see there are cremaster muscles here. There's dartos muscle, two kinds of muscle, the cremaster muscle and the dartos muscle. Cremaster muscle basically is a small bundle skeletal muscle which is in continuation with the internal oblique muscle. It elevates the testes during the sexual arousal or exposure to cold. So it pulls the testes up towards the body. The dartos muscle is basically smooth muscle fibers. It causes the wrinkling of the skin of the scrotum. When we are discussing the cut section of the testes, so you can see this is the testes. It is divided into various lobules. So this is the tail of the epididymis here. You can see the septum that separate one lobule from the other. Inside, you can see there is this the ductus epididymis. From ductus epididymis, various tubules. This it would keep on dividing to form other parts. So you can say that this is the tunica albuginea. This separates each lobule from one another. There are seminiferous tubules. So these are the seminiferous tubules. These seminiferous tubules are present. These are the ones where basically they are about 200 to 300 lobules within one testis. So spermatogenesis occurs here. Spermatogonic cells are present here. And interstitial endocrinocytes, the interstitial cells of Leydig cells are also present here, which release the male hormone testosterone. So you need to keep in mind the labels: the seminiferous tubule, the tunica vaginalis, tunica albuginea, the lobules here, the septum, the tail of epididymis, the ductus epididymis, the retitestis. This bunch is called the retitestis. The body of the epididymis is here. This is the efferent duct, and this is the ductus deferens, and this is where the spermatic cord attaches. This is how the sperm goes out of the testes from this particular part. So if you cut this in section, you see these are the Leydig cells present in between them. And this structure, if you magnify it, you will see Sertoli cells. Sertoli cells are present here. These are spermatids in which the tails are still growing. So the spermatogonia, they are lying next to the basement membrane. The sperm cell, the spermatozoa, it reach maturity and it burrows, it heads here into the lumen. So you can see this is region here. This is the lining, the basal lamina. There are the spermatogonia here. They will divide to form the primary spermatocyte. The primary spermatocyte then converts into the secondary spermatocyte. The secondary spermatocyte turns into spermatid and the spermatozoa. And these are the tails growing from the spermatozoa. So meiosis is occurring actually. If the spermatogonia they divide by mitosis, then it starts to undergo changes. So primary spermatocytes are formed, then secondary, and then spermatozoa. So by the time spermatozoa is formed, it's basically the meiosis have completed. Between the developing sperms are in the tubules called sustentacular. They are fertilized cells embedded in between the developing sperm. These sustentacular fertilized cells they nourish the sperms. they mediate their effects of testosterone and FSH on the spermatogenesis. They phagocytize the excess is permitted cytoplasm. They are also secreting fluid which is rich in estrogen, androgen, and potassium, glutamic acid, aspartic acid. Internal to the basement membrane are tight junctions that join the fertilized cell with each other and form the blood testis barrier. Spermatogenesis is shown to you here. There will be a diploid cell called spermatogonium. It divides to form it will just grow on to form the primary spermatocyte. The primary spermatocyte undergo meiosis, so it turns into secondary spermatocyte. Two cells are formed having haploid number of chromosomes. They will then further form four different cells by meiosis two. So spermatids will be formed. So these spermatids will not have any tails, so they need to they will burrow their heads here and they will keep on growing the tail. So their spermiogenesis occur and the mature spermatozoon will be formed. A mature spermatozoon is having two main parts. There's a head and there's a tail. The head is having acrosome, which is having the enzyme to digest the egg wall. There's a nucleus, which is haploid. There's a neck containing mitochondria and the middle piece. And then there's the principal piece, which is the tail. That may it varies from one sperm to another how long the tail is, and then there is an end. The ductus testis, ductus epididymis. This is how the ducts are present here in the testes. So let's discuss the ducts in detail. Once the spermatozoon is released into the fluid from the seminiferous tubule, they enter the straight tubules and then they go to the reti testis. From the reti testis, they leave into the epididymis and then into ductus epididymis. The ductus epididymis, the epididymis is basically a comma-shaped organ. You can see that this is the comma-shaped organ lying at the posterior border of the testes and consists of tightly coiled tube called ductus epididymis. The ductus epididymis is lined with pseudo-dorsified columnar epithelium. Functionally, the ductus epididymis is a site for sperm maturation. It takes about 10 to 14 days for the motility to increase and become fertile. It stores spermatozoa for at least a month and helps them propel the spermatozoa towards the urethra by peristaltic contraction of the smooth muscles. The ductus of testis continued. The tail of epididymis is less convoluted as it goes out. It forms the ductus vas deferens or the seminal duct. It also stores the sperm that are conveyed to it through the epididymis. It ascends to the posterior border of epididymis. The dilated portion of this vas deferens is called as ampulla. The spermatocord supports the structure of the testicular artery. All these structures are supported there. There is an inguinal canal which passes through this and originate deep into the abdominal ring. So this is actually the where the testes are made when the individual is a fetus, and they descend into the scrotum through the inguinal canal or the inguinal canal. The canal ends at the superficial subcutaneous inguinal ring. The ejaculatory duct of urethra is posterior. It is posterior to the urinary bladder. The ejaculatory duct ejects spermatozoa into the prostatic urethra just before ejaculation. The urethra is a terminal duct of reproductive and urinary system, common for common pathway in men. It is divided into three parts: prostatic urethra, membranous and spongy or cavernous urethra. Prostatic urethra passes through the prostate gland. Membranous urethra is a muscular portion between the two ischial and the pubic rami. The spongial cavernous urethra passes through the corpus spongiosum of the penis. Spongy urethra enters the bulb of the penis at the end, the external urethral office, which there is a terminal slit-like opening, which is formed by the enlargement of the distal urethra with the glans penis within the glans penis. So the accessories, now we come to the accessory sex glands and the semen. The accessory sex gland include a paired seminal vesicle. The seminal vesicle include alkaline, they basically secrete alkaline fluid which helps to neutralize the acidity in the female reproductive tract. So we release viscous fluid which contains fructose. ATP is required for the production of sperms as well as for motility. Prostaglandins are there, which promote sperm motility. Sperm motility. Fibrinogen helps the coagulation of semen after ejaculation. So once the semen is ejaculated into the female reproductive tract, because of fibrinogen, it will start to clot. It will stay coagulated for about 25 to 30 minutes, and after that, the sperms they start to move because the clot then dissolves. So fluid is secreted by seminal vesicle. It constitutes 60% of the volume of semen. So this is giving the largest contribution to the semen, the seminal vesicle. Prostate is a single donut-shaped gland. This is inferior to the urinary bladder and surrounds the superior and posterior portion of the urethra. It secretes slightly milky, acidic fluid and contains citric acid and enzymes such as phosphatase clotting enzymes that helps the fibrinogen to coagulate the fibrin as well as fibrinolysin. So fibrinolysin start to break the clotted coagulation. So within 5 to 20 minutes, the clotting they start to break, and by 30 minutes, the semen is almost liquefied back. There's a bulbourethra. The secretion of prostate basically adds 25% of volume to the semen. Bulbourethra corpus, that is the neck, it lies at the base of prostate gland. It secretes alkaline substance. It is required for the sperm neutralizing the acidic environment of urethra, because the urine is actually always, in most cases, is acidic. So there is a mucus lining that lubricates the lining of urethra, decreases the number of spermatozoa that can be injured during ejaculation. Mucus also lubricates the end of the penis during sexual intercourse. Thus, semen, which is about in volume in about 2.5 to 5 ml, with 50 to 150 million sperms per millimeter in ejaculation, in an ejaculate, are released in an ejaculation. The sperm, they secrete an enzyme hyaluronidase and proteases. The proteinases stay from the acrosome to digest the covering of the ovum, and has a slightly alkaline pH, 7.2 to 7.60. Semen also contain an antibiotic which is called seminal plasmin, and they have the ability to destroy certain bacteria. Coming to the details of penis, penis is a cylindrical shape and consists of a body, root, and a part called glans. Penis is used to introduce sperms into the vagina. The body of penis is composed of three cylindrical masses. Each is surrounded by a fibrous tissue called tunica albuginea. There are two dorsolateral masses called corpora cavernosum in the penis, and the central mass called corpus spongiosum in the penis. All three masses are enclosed within the skin and fascia. There are vascular changes which result in as erection of the penis, which allows ejaculation and the propulsion of the sperm. This is basically a parasympathetic reflex. The root of penis is attached to a portion called the bulb of penis. That expanded portion of bus called as the corpus spongiosum of the penis. There is a region called crura or crus of the penis that separate and tapered portion of corpora cavernosa penis. The distal end of the corpus spongiosum penis is slightly enlarged, acorn-shaped, and called as the glans penis, the margin of which is travis corona. So you can see this diagram here. In this diagram, you can see the prostate gland, then there is an ejaculatory duct passing through it, there is the prostatic urethra. The prostatic urethra then passes through the bulbourethral gland, and this is the crus of penis, which is present near the bulb. The bulb is in the central region. Then there are the corpus cavernosa penis, which are, you can see by the sides of this. There are deep arteries in the penis which are required for pumping blood into these tissue, which is required during the erection of the penis. Then there's corpus spongiosum, which is more lateral, which is more midline. So corpus cavernosa is on the sides, and corpus spongiosum is towards the center, and the spongy urethra is the lining which is present in the center, in the midline, through which is the passageway for the urine and semen to pass through. The corona is the crown-shaped region present at the end of the penis. There's a region called glans penis and purpose. This is the foreskin which is removed during circumcision in Muslims and in Jews. The hormonal testicular axis involves gonadotropin releasing hormone released from the hypothalamus. It stimulates the anterior pituitary gland which is having cells called gonadotrophs. They release FSH and LH. FSH is involved in initiating the spermatogenesis. It goes on to act on the sustentacle or the certilis cells in the seminiferous tubules. Then LH, it stimulates the release of testosterone secretion by the testicular cells, the Leydig cells. The Leydig cells are the one that release this testosterone. So testosterone is then converted into dihydrotestosterone as well. So dihydrotestosterone is required for the male pattern of body development before birth and as well as after puberty. So it includes all the male secondary sexual characteristics. They all come because of this dihydrotestosterone, which is a derivative of testosterone. The negative feedback of testosterone control in the same way as all negative feedbacks are controlled when the Leydig cell, the testosterone, they are stimulated less. So a lower level of testosterone will be there in the blood. It will cause the homeostatic balance to shift in the favor of GnRH, which then cause the GnRH to be released, which in turn cause more FSH and LH to go in the blood, more FSH to go in blood, and because of that, the testosterone level will start to increase. That's all for now. Thank you.